Good morning, everyone, and welcome to Denver Museum of Nature and Science at home. So excited to have you all joining us and seeing from all the places around Colorado and beyond that you're tuning in from. So welcome to our COVID-19 webinar series. Every Monday morning, we've been trying to bring you this series bright and early at 8:30, where we're trying to share the science and the stories that matter. So we're just really excited to have you tuned in. Thank you to all my museum colleagues behind the scene to helping us put this together. And a huge special thank you to our partners in all this, the Colorado School of Public Health, for helping us bring this webinar series to you. I'm Kristen Uhlenbrock. I work at the Institute for Science and Policy, a project here at the Denver Museum of Nature and Science. So just a couple housekeeping items before we jump right in. I think many of you are familiar that we're hosting this both here on Zoom as well as on Facebook Live. Hello and welcome to both audiences. As Nicole was telling you to open that chat and introduce yourself, that's a feature here on Zoom where you can ask and type in your questions. We encourage those throughout the presentation. We will have a Q&A portion towards the end. I will try to incorporate as many as I can, and I just want to give the disclaimer, of course, that we see them, we hear them, we read them, we appreciate them, and we do not get to answer and ask all of them, but please keep them coming in. If you're tuning in over here on our Facebook Live channel, welcome over there. Just go ahead and use that comment feature to ask in your questions for our audiences, and we'll take those into consideration as to and bring them over. So today's episode is all about zoonotic diseases, the spread of germs between animals and humans. COVID-19, of course, is a zoonotic disease, and it isn't the first to make that leap, and it sure as heck is probably not going to be the last. Y'all are probably well aware of other very well-known and high-profile zoonotic diseases such as Ebola and SARS and Zika, to just name a few. A lot of these diseases come about because of that close connection and interconnectedness that we have between the animal world and the human world. And so our speakers today are going to give us a little bit of their perspective on that disease transmission and better transmission pathways to understand and think about how did COVID-19 emerge, as well as how do potential future emerging threats come about so that we can be well aware of those. We're also going to hear a little bit about sometimes how these animals that have these diseases, which there are numerous of, don't get sick, right, or appear healthy. And so what does that look like? So I feel very honored to have two of our research professors right here in our own state of Colorado from Colorado State University joining us. Both these professors are in the Department of Microbiology, Immunology, and Pathology, and I actually think they're office neighbors who sit just kind of right next to each other down the hall. Up first is going to be our speaker is Dr. Brian Foy. His research spans both across basic and applied biology, focusing on the interactions of vectors with their hosts and with vector pathogens. He has really good diverse interests across the board, and it's been reflected in his training throughout his career, where he did his undergrad in Notre Dame in medical entomology, anthropology, and ecology. And then he went on to grad school at Tulane, where he did molecular and cellular biology, immunology, and tropical medicine research. I think that's where he received his PhD. His current interests lie in defining concepts that govern blood meal acquisition and digestion by vectors, and parasite and arbovirus transmission from vertebrates to vectors and vice versa. Dr. Foy is also keen on using that knowledge combined with epidemiological concepts that define vector-borne diseases to practically control their transmission. Our other speaker, our second guest speaker, is Dr. Tony Schounts. His research interests are in understanding immune response of reservoir hosts, particularly of that of rodents and bats, to understand emerging zoonotic viruses that typically lead to apathogenic persistent infection in bats. So in other words, bats that get and carry infections, but not the disease. His lab is also interested in the role of immune response during infection and vaccine development and therapeutics for zoonotic viruses. So his lab looks at why bats hosts can have such a high impact human pathogens that cause no discernible disease in the bats. He actually has a unique carrying colony of, I think, Jamaican fruit bats in his lab that I think we may see some photos of today. Dr. Schounts received his PhD from Kansas State University. So with that, welcome to everyone today. We're going to take a step back and just take that big picture view about zoonotic diseases and the spillover effect, and take a quick look too and talk about how COVID-19 got transmitted and emerged. So let me introduce first Dr. Brian Foy. Good morning, Brian. How are you?
Great. Thanks for inviting me, Kristen. I really appreciate it. And as well, appreciations to the Denver Museum of Nature and Science and the ISP and the Colorado School of Public Health for inviting me. All right, should I share my screen now? All right, so, apologies, here we go. I'm hoping y'all can see that. I'm gonna put it on presenter view. And I'm gonna give you the broad overview about how zoonotic pathogens spread amongst animals and people. And then I think Tony's gonna get a lot more specific with COVID-19. So I'm starting out with this nice slide by the CDC, which shows very simply how zoonotic diseases, which are caused by zoonotic pathogens, most often viral pathogens, are spread between animals and people. I think the most important part of this slide really is that you see arrows going both ways. And this is really important to know because we, as much as animals can spread things to us, we can spread them back to animals. And of course, that's because we are animals and we have evolved to depend on many non-human animals as well as they have evolved to depend on us. And so the simple answer to prevent the spread of zoonotic diseases and pathogens amongst humans is not just to get rid of the certain animal species that is spreading a pathogen to you, because of course the animals provide many benefits to us. We're all part of one planet. And we need to think about this. And this is even important for those bats that you see on the right-hand side. Bats are thought of as farmers of the tropics. They fertilize many, many plants, and they are really good at seed dispersal. So even though you might think of bats as scary, they're not, and they're really helpful to humans. But nonetheless, more than 6 out of 10 every known infectious diseases can be spread from animals to humans. And we think about 3 out of every 4 new emerging infectious diseases come from animals. In fact, this is probably an underestimate. And even the pathogens of humans that probably don't spread anymore from animals once did. For example, I work on malaria that's transmitted or that's caused by Plasmodium. And Plasmodium, many of those species are not shared amongst other animals or only humans, but that's because they've just adapted to humans over hundreds of thousands of years. That spillover event that happened with Plasmodium probably happened from primates hundreds of thousands of years ago. And since then, it's just simply adapted to humans specifically. So pretty much all diseases, infectious diseases can probably have their origins, if not recently, from other animals. This is a nice article from The Lancet that talks about, that kind of gives a nice graphic about how these things spill over and ultimately spread in humans, these zoonotic pathogens. You can see on the right, I'm gonna make my pointer bigger. On the right here, you see these wild animals that are kind of sharing zoonotic pathogens. And spillovers, in fact, happen even amongst themselves, different species. For example, like a rat might spread into a monkey. Maybe that monkey caught the rat and is eating it. And you might have a pathogen that spreads from that rat to the monkey. But we just don't see these spillovers. And maybe it causes a disease in the monkey population. But we don't see that because it happens out in the wild. It happens in nature somewhere. But every once in a while, these spillover events, as they cycle between wild animals, eventually spill over into humans. Most oftentimes, they spill over first in the domestic animal reservoir that we have very close to human populations. We're talking about animals that we use for agriculture, we keep as pets. So camels, cows, chickens. And oftentimes this spillover happens when we directly, the wild animal is directly interacting with the domestic animal, or a vector, as you can see here, for certain zoonotic arboviral spread pathogens can bite one of these and then transmit it to the others. Then nonetheless, a spillover event occurs. You have amplification in the domestic animal population, and then another spillover event occurs and to cause disease in the human population, these human cases here in red. So how do we deal with that? It shows nicely on the right-hand side that there's many ways that we should be dealing with it if we're doing a good job of this. The first is just forecasting readiness. This is monitoring animal populations in the wild, monitoring outbreaks and diseases in them, seeing how close we are encroaching onto them or they are encroaching onto us. And then early detection signals, which is up here, which would be our surveillance efforts, if they're funded well, amongst animal populations that are wild as well as domestic, to see how often a spillover might occur or has already occurred and is just in the early phase of outbreak and epidemic before it reaches pandemic proportions right over here. We should also have control operations ready to go, which includes everything from vaccines that are very broad to target many different viruses that might cause these pandemics, to therapeutics, to control tools for controlling vectors so they can't bite and spread pathogens to us. And as well, finally, a rapid response, which would be everything from public health measures that we have ready to go to just really flatten this curve, this red curve for the disease in people. Now, these pathogens, we've been seeing an increasing number of them ever since maybe the 1950s. This on the right, on the left, I'm sorry, you see a cumulative number of pathogen species that are emerging in people over the last several decades, HIV being in 1980s. This is the pathogen that's not viral that causes mad cow disease. It's actually a protein, a prion protein. And then SARS, which occurred in 2002, 2003. A disproportionate number of these emerging pathogens, zoonotic pathogens, are indeed viral species. And you can see the rapid rise of new viral species accumulated in people since the 1950s. And there's many reasons for that. The main one being viruses are incredibly diverse and incredibly numerous. There was a nice paper, actually a book, in fact, that was published by the Institute of Medicine out of the National Academy of Sciences, right around when SARS-1, the first SARS virus, emerged in 2003. And this outlines at least 13 different reasons why pathogens emerge from zoonotic reservoirs and into people and cause pandemics, and kind of gives broad outline. And I'm going to go over some of these very briefly to kind of give you a feel for how these occur. So the most obvious one that everybody thinks about at first is microbial adaptation, or we can substitute viruses if we're talking about SARS here. Virus adaptation, change, mutation, natural selection, and evolution. And there's another paper published several years ago that kind of really outlined this early event of microbial adaptation and change. And they kind of break it down into 4 different events. The exposure of the new animal species, in our case people, to that zoonotic pathogen, the infection that ensues, the spread that then spreads from that initial patient 0, for example, and ultimately the adaptation of the microbe to many people as it spreads. What I think that this should probably be more clear is that they show this as a very linear graph from exposure, infection, spread, and adaptation. But in fact, it's not. And they cycle back around, because even on that exposure event, you might have a mutated virus that allows that exposure or that infection event to really establish. And so this isn't linear, but in fact, a lot of these different connect over cycles of virus transmission or pathogen transmission to people. So when you first think about that exposure event, really what you need to think about is how closely related are these animals to the spillover recipient animal. And let's just say that's us, humans. And of course, humans are primates right here. This is a phylogeny of mammalian orders across the world. And it basically shows the relatedness of different animals, mammalian animals. So all the reds are all more closely related than all the ones here in purple, and we're in the purple here. So we're more closely related to these Dermoptera and Scandentia, which are kind of unique, odd mammals, but we're more closely related also to rabbits and to rodents than we are, for example, to marsupials and that you see most often in Australia. So the relatedness is shown here, but also the numbers of species. And you can see very quickly that rodents are extremely numerous as a species, as are the Chiropterans, which are bats. And so the first thing you should be thinking here is the most easiest thing to spill over into us primates are things within our order, as well as what are we interacting with most frequently, and how numerous are these animals. And that's why we see a lot of these zoonotic pathogens, in fact, as rodents, rodent-borne, and chiropterans, even though they're not as closely related, there's many, many species of them, and they're very interacting, they're very overlapping with us in ecology. We see many, many bats, especially in tropical areas, and highly dense tropical areas of cities, like Rio de Janeiro or something, many, many bats. We also think about, during that infection event, how easy is it for a pathogen to spread from a donor species, the wild animal, to us, the recipient species? You can either have a very steep fitness valley, meaning a very deep valley, which is hard for that pathogen to jump from the donor to the recipient because we are so different, for example, from a marsupial. While that fitness valley might be much shallower, if we go back here and we see that it came from a lagomorph or a rabbit, because we're much closely related to rabbit. And if a virus is replicating in a rabbit, maybe it has an easier time being able to replicate in our cells because we're closely related to it. So there might be a shallow fitness valley there compared to a steep fitness valley. We also think about spread during this microbial adaptation and change. So this is a really good example from Wikipedia showing two different strains of influenza, H1N1 and H5N1. Certain strains can spread much easier amongst people because of what their pathology is in our certain cells. H1N1 tends to have a predilection for cells of our upper respiratory tract. Now, this doesn't mean that it's as fatal as H5N1 because it's not really affecting those deep lung tissues, but it is much easier spread in coughs and sneezes. And so it's spread around much more quickly than H5N1, that really lethal pandemic influenza virus that we had, but which is mostly in our lower respiratory tract and is not spread as quickly, even though it's more fatal. Unfortunately, as Tony will probably talk about, coronavirus, the COVID-19 that causes COVID-19 is probably a good combination of both of these. And then finally, a lot of people think about mutation as well as reassortment and recombination under this adaptation. So viruses are quasi-species, they replicate like crazy, and they don't have good proofreading mechanisms or any proofreading mechanisms of when they replicate their genomes or their genetic material. And so when we think about mutation, we think about these things replicating very quickly, not having good proofreading compared to, for example, us when we replicate our own cells have for their genetic material. There might be a mutation, like as shown in this star, that ultimately manifests itself in a changed protein on the surface, like the receptor of the surface of coronavirus. We see already mutations in the coronavirus genome, which has caused mutations in the receptors that allow them to now bind people's cells in people much more efficiently. But viruses also undergo reassortment and recombination. Reassortment is very common, what you see with influenza, and that's why we have to have a vaccine every year for influenza. Because when two different viruses, as you see here, infect a single cell, their genomes, which is in many segments, can reshuffle like cards in a deck. And so you can see here, this reassorted virus has gold as well as blue genome segments, which can give you a completely different strain. We can also have recombination events where a splicing event can occur between one part of the genome. For instance, in this sample, a blue and a yellow genome made one part of the genome and that encodes for the receptor protein being a recombinant or a mutation that really splices two different viruses together. We see this, evidence of this with SARS coronavirus 2, or that causes COVID-19. There are many others besides microbial adaptation, these different factors. A lot of people think about climate change. This is from the Intergovernmental Panel on Climate Change, the predictions about what our Earth will look like in many years relative to the recent past. Not only, this is a very complicated factors that drive, they certainly do drive emergence of zoonotic pathogens, but it's complicated because while things are getting warmer all across the globe or predicted to get warmer, it's also predicted to get Some places very much more wet, while other places much more dry, like the desert Southwest. Some pathogens thrive in dryness. Some pathogens thrive in precipitation or a lot of moisture. And so which pathogens arise over time because of climate change or changing ecosystems will really depend on the type of virus or other pathogen and where it's emerging in the globe. International travel and commerce is a huge one that is really can't be underestimated. We have been running across the globe for many hundreds of years now, but it has really taken off even since the 1980s, 1990s. How much air travel there is just incredible, and it's because human populations are so much more mobile. They're becoming more wealthy. Before, flights were the realm of rich people. Now, many middle-class people can take flights, and a lot of people in very previously poor areas can even jump on a motorbike, as you saw with Ebola in West Africa spreading around. This is a screenshot of how much air travel happens across the globe on any given day. It's amazing how quickly this can happen. Arboviruses that are transmitted by vectors are most often transmitted by just travel and introducing either the mosquito or tick into new places or the infected vector. And we think this is probably what happened with West Nile virus introduction to the United States in 2001, probably why Zika virus had a large pandemic in 2016 and ultimately spread from the Pacific South Pacific into South America, and then spread from there as it got into new mosquito species in the Americas. And on the backside of this international travel, they pair it with commerce. And of course, commerce has been a big driver of the spread of zoonotic diseases. The most obvious one everybody thinks about with SARS coronavirus is the wet markets that seem to act as at least amplification, if not introduction, into very large human populations in large cities in China. So these wet markets were key for the explosion of SARS-CoV-2, and there's debate about how much they really mattered in SARS-CoV-2 that Tony will talk about as well. So when I say wet markets, we're talking about the keeping of exotic wild animals in these open air markets and the immediate slaughter of them for fresh meat for people that would like to consume them. There's also agricultural practices can really drive the spread of zoonotic agents. We think about this specifically with Nipah virus, which is one of the henipaviruses in Southeast Asia. The agricultural practices of keeping pigs and pigsties underneath fruit trees, where a lot of bats, who are natural reservoirs of these viruses, were eating the fruit above the pigsties, eating some of it, salivating into that fruit. Maybe that fruit dropped and then the pigs ate it. Then the pigs got infected. It ultimately spread it to the farmers that were the keepers of the pigs, and that caused spreads and big outbreaks of Nipah viruses. And now, even though we've kind of cleared up this in Malaysia, these practices, the spread keeps happening in places like Bangladesh because there are date palm sap workers who collect sap at the top of trees, and infected bats seem to be licking that sap and then infect people that don't pasteurize that sap. And then finally, I'll just end with this one: lack of political will. It's pretty clear by most political scientists and scientists that the lack of political will really drove the spread of the HIV epidemic in the early 80s. And unfortunately, we're seeing a bit of evidence of that today and haven't learned our lesson from 200 years ago, where there were people having anti-mask meetings and protests back in the Spanish influenza epidemic in 2018, and we're seeing the same thing 100 years later. So I will stop there and I will share it to the next speaker, Tony.
Great, thank you, Brian, very much. Very informative. We're gonna have to come back and ask you some questions specific to that, but let me turn this over to Tony. Good morning, Tony. How are you, and welcome. Tony, let's take you off mute. Start that over.
Good morning. I'm doing okay so far, but the day is early. So Brian did a nice job of setting me up, and so I'm going to try to keep up the pace and clarify some points, at least from my perspective, about how some of these zoonotic agents have become really important in terms of not just public health, but also how these outbreaks can really cause other consequences, including impacts to our economy. So my interest is in zoonotic viruses that originate in mammals. Brian principally studies mosquito-borne diseases, and those are really important in terms of global health. But my history has been principally involved with deer mice and hantaviruses. So those of you who live in Colorado, you should probably know about hantaviruses because it's a real problem. And then of course bats and their status as reservoir hosts for a number of other infectious diseases. And so I want to first start out with this next panel, if I can get my pointer going. These are a number of infectious diseases that are all zoonotic diseases, right? So they originate in animals and somehow manage to get into people. And each of these diseases is caused by one or a few number of viruses. For example, Marburg virus is caused by a virus called Marburg virus. But then we have Ebola virus disease, which is caused by a number of Ebola viruses. And so even though there are 3 viruses, they end up causing the same disease. And I just want to point out that what's interesting is Marburg and Ebola are very close cousins to one another. And the very first case of Marburg virus disease in the United States was diagnosed in Denver a few years ago in a woman who had traveled to Africa and had managed to contract it there. So each of these viruses cause these different diseases. And of course, here are hantaviruses, and there are dozens of hantaviruses that can cause hantavirus disease throughout the world. Brian's already talked about Nipah encephalitis, and that's caused by Nipah virus. But if you look at the animals that host these viruses, most of them are either bats or rodents. There are a few oddballs down here. For example, hepatitis E, swine, wild deer, and boar. But most of the rest of these are hosted by rodents or bats of some kind. And I'm going to talk mostly about bats today. And so this picture here, I took when I was flying from Kisumu, Kenya, a few years ago on my aircraft out of Kisumu. And I took this photo because it puts into stark perspective why these zoonotic diseases get an opportunity to get into humans. And so you have societies that have sustenance farming, and so you have a home here, and then crops planted out here, which attracts wildlife. Oftentimes, these people will have, for example, chickens in their yard, or sometimes swine, and that introduces contact between the wildlife and the domesticated animals, which then end up transmitting the viruses to the humans, which can then lead to outbreaks of infectious diseases. And so this is one important way. There are others, of course. We can always have accidental events where we have spillovers that occur. So what I'd like to do today is mostly talk to you about bats and their viruses. So far, there have been thousands of viruses that have been detected in bats. Now, I want to emphasize that probably the great majority of these viruses are incapable of causing disease in humans. But like any statistically unlikely event, if you do something enough times, then a statistically unlikely event becomes statistically likely. And so considering that we know of thousands of viruses in bats and probably thousands or tens of thousands of more that we don't know about, then this is going to be an ongoing and persistent threat to human health in the coming decades. Now, I want to first talk a little bit about the viruses and how they reside in their reservoir hosts. And if you're a virus, right, you are completely dependent upon the life of that animal that you're in for your own livelihood. So if you want to persist as a virus as a viral organism, then you either have to infect your reservoir without causing disease or eliciting a sterilizing immune response. If either of those events happen, then bad things are going to happen to the virus as well. So there's a second way of doing that. You can simply get out of the host before you cause substantial pathology or a sterilizing immune response, and then jump into a next susceptible animal that can then set up the second round of infection. And so these are the two strategies that most viruses take when they're trying to exist in nature and to spread. Because viruses are just like all other biological entities. They have to figure out a way to replicate and have their offspring passed on and to complete their life cycles as well. So what has typically been observed is that in most of these infections, the first one is what happens. You rarely see disease in the reservoir host from these viruses. The viruses infect them. Sometimes they have abundant virus replication early on, but then it sort of subsides. And then the virus can sort of persist for weeks or months or even the lifetime of the animal, with periods of where virus reemerges and then gets shed into higher amounts. And then that can then sustain the life cycle of the virus by going from, for example, one bat to the next bat to the next bat to the next bat. When these viruses get into humans, that's a bad thing for the virus as well, because that's generally a dead end for the virus, because most of the time these viruses don't efficiently transmit from person to person. That's not always the case, as we see with SARS, for example, COVID-19, for example, and other infectious diseases. But generally speaking, these viruses want to stay in their reservoir hosts. So what we think about in terms of these viruses and their relationship to humans is that if you want to understand how these viruses behave and how they get into humans, then you have to understand the ecology and the biology of the viruses. You can't just study the virus, you have to study the reservoir host that it's in. And unfortunately, most laboratory scientists never set foot out in the field. They spend their entire careers in a laboratory wearing a coat or a biosafety suit, and that's what they do. They don't actually go out and do field work where they can then really understand the biology and the ecology of these viruses in the context of their reservoir species. Okay, so when we go out in the field, we have to take some precautions. For example, in this upper panel here, this is a custom-built biosafety cabinet that I had made many years ago. What it does is it allows me to plug it into the cigarette lighter of a truck, and it runs a motor, electric motor that pulls air in and then sterile filters it, blows it into this container, this area here, which is what I'm doing right here. And then I can process in a sterile environment that also protects me from getting any virus that this particular deer mouse might be harboring. And then at the end, it blows the air out the other side, where it goes again through an air filter, a HEPA filter, to a sterile filter, and then blows the sterile air out. And so these are the kinds of things that we have to do to not only preserve sample integrity, but also to ensure protection of us. We also use special personal protective equipment to help process animals in the field as well. And here's a picture of a long-tailed pygmy rice rat, which is a hantavirus reservoir in South America from a field study that we've done before. Okay, so we're all interested in bats right now because of the viruses that they carry. And this is just a short list of the ones that are really important bat viruses that when they get into humans cause a substantial disease. Rabies virus and other lyssaviruses. Most rabies cases in the United States now are from bats, and that's not because bats are suddenly more important. It's because we've gotten rabies in dogs and cats under control, and skunks and raccoons to such a great extent that there are very few cases that come from those animals. But then the paramyxoviruses include a Nipah virus that Brian just mentioned briefly, but a couple of others. It's now pretty clear that mumps virus, which is one that we vaccinate our children for, is probably a bat virus, and it circulates in bats in certain parts of the world. And of course, that now makes us rethink the chances of us eradicating this virus. If you want to eradicate a virus by vaccination, for example, as we did with smallpox, then that virus should only infect humans. If it's a zoonotic virus, that really dampens your enthusiasm for trying to eradicate that virus, because you can eliminate it in people, but you could always have a future event where it spills back over and starts over again. And then, of course, we've already talked about the Ebola viruses and the Marburg virus. Of course, Ebola is the most famous, I think, until now. And then the coronaviruses. Just in the last 2 decades, we've had 3 really important coronaviruses that have emerged that have affected humans. And what's the biggest issue for many of us is that these viruses, most of them either require biosafety level 3 or biosafety level 4 containment. And those are the 2 highest levels. So here at CSU, we have BSL-3 containment, but we don't have BSL-4 containment. So we can't work with Nipah virus or Hendra virus or the Ebola viruses, but we can work with all 3 of the coronaviruses because they're considered BSL-3 viruses. And if any of you saw that movie Contagion, then you know about the origin of the virus that was in that. And that story was about Nipah virus that had undergone a recombination event that Brian mentioned earlier with an influenza virus, and that made a sort of a hybrid virus that had genes from 2 different groups of viruses. And so if you remember that movie, you may recall that. If you haven't seen this movie, I encourage you to do so, because scientifically it's pretty accurate. Most of these movies that we see today are really disappointing to me as a scientist because I know it's mostly BS. But this movie in particular was actually pretty good in terms of the fidelity to the science. Okay, so getting out and doing field work has a number of challenges, and I won't read this list. You can look at it if you want, but there are several things that are really challenging for us. For example, if you're going to go into a cave that's loaded with bats, here's a bat carcass right here, then you have to have all this personal protective equipment on. So you've got a covering over your body. You have leather gloves that are duct taped to your latex gloves that are underneath. Have to have a respirator on so that you can filter the air out to make sure it's sterile air that you're breathing in, because who knows what viruses are floating around in the air here. When you're in a cave, you want to wear a hard hat because you can't see, and you'll bang your head on this, and that could introduce a cut that could then lead to an introduction of an infectious agent. This person has taken his helmet off just for this picture. So the light that you see is from a camera flash. Otherwise, it's completely dark in this cave, and the only light is from your headlamp. And if your headlamp fails, then you can't see where you are, and you will not find your way out unless you happen to also bring a lifeline with you that you can grab a hold of and follow your way back out of the cave. That's what keeps you from getting lost in that cave. So here's another picture down here of all the bats that are found in this particular cave, and they're in very high density, and so these are the sort of the parameters that we have to deal with when we're working in some of these environments, trying to find where these viruses are in their natural environments. Okay, so let's just briefly talk about coronaviruses, and then I'll end it on this slide. So it's pretty clear now that probably all coronaviruses originated in bats. And as I mentioned earlier, there have been thousands of viruses that have been detected in bats, and more than 1,000 of those viruses are coronaviruses. Again, these are the ones that we know about. There are probably thousands more, perhaps tens of thousands of more that we don't know about that are circulating in bats. There are about 1,200 species of bats in the world. They're the second largest group to the rodents, which have about 2,200 species. And so that's, as Brian pointed out, why we're really interested in bat viruses and rodent viruses, simply because of their high density and this feature that many of them have that's called peri-domestic behavior. In other words, they don't really have a problem coming into your house or near your house or on your property. And when they do, they're usually looking for food. Sometimes they'll build nests, and if they have viruses, they'll bring those viruses with them. And that then puts you at exposure risk for contracting some of these viruses from these animals. Now, of those thousands of coronaviruses that have been detected in bats, we've detected several hundred that are SARS-like coronaviruses, right? And already we've had 2 emerge SARS-like coronaviruses that have emerged in just the last 20 years. Before 2002, the coronaviruses were really only known for causing mild colds in humans. Occasionally, they could cause serious disease in people who had immunodeficiencies or were under because of chemotherapy or HIV disease, but generally speaking, they were rather innocuous infections. Then in 2002, we had the first outbreak of SARS, severe acute respiratory syndrome. And then 2012, and that virus went away within a year, right? And the reason it went away is because it doesn't transmit very efficiently person to person. But more importantly, people show signs of disease before they start shedding the virus. And if you have that situation, then it's easy to control. You just quarantine those people until they get better. And that way you can knock the virus down. And that's what happened with SARS. That's not what's happening with SARS-2. The second coronavirus was Middle East respiratory syndrome that emerged in 2012. That virus hasn't gone away. What it's done is a little bit unusual. It's established itself in what we call a secondary reservoir host. So even though it originated in bats, it now circulates in dromedary camels much in the Middle East. And every year we see outbreaks of MERS coronavirus infection, and it's always coming from camels and into people, not from bats into people. But the original transmission event was from a bat, we don't know the species, but from some species of bat into camels, and now it's spread throughout camels in North Africa as well as the Middle East. And now we're faced with, briefly, just another one that's an animal health issue, swine acute diarrhea syndrome, which causes the Chinese swine industry billions of dollars in lost revenue. That was also a coronavirus that emerged from a bat. And then now we have 2019. The disease is called COVID-19, And the virus that causes it is SARS-CoV-2. So SARS-CoV-2 emerged, and unlike SARS, it not only efficiently transmits person to person, but there are a number of people who are asymptomatic, completely asymptomatic, so they get infected and never show signs of disease. And then there are others, virtually everyone else, who are asymptomatic for the first few days, yet they're still transmitting the virus. And that makes it very difficult to control, because those people go about their daily lives and they don't feel sick, and so they go to work and they go hang out at bars and places like that, and they transmit the virus without knowing they're infected. And that's what makes this virus particularly challenging. If we could get 95% of people to wear masks and keep that 6 feet distance, and make sure that we limit the size of groups and follow all of these distancing practices, we could probably get this virus under control in a couple of months. So then the question is, when will the next virus emerge? And I don't know when, but we've had 3 human coronaviruses emerge in the last 20 years. I think it's going to be sooner rather than later, and they're all going to require different strategies for control and different types of vaccines and drugs to treat them. So just as Brian mentioned earlier, China is a much more mobile society today in 2020 than it was in 2002. And along with that, human mobility increases the chance of spillover events happening in the future. So I'm certain that there are going to be many more coronavirus spillover events in the future, unless we take really, really dramatic steps to limit the chance that a zoonotic virus can get from a bat into a livestock species or into humans. Otherwise, I think this is going to be a continuing process. What is pretty clear now is that although SARS-CoV-2 originated in a bat, this is a Chinese horseshoe bat, it probably got into a second mammalian species that we don't know, where it underwent one of those genetic recombination events that Brian just talked about, that then allowed it to efficiently bind to human cells, and that is what probably initiated this outbreak. I personally don't think it started at the Wuhan wet market. As Brian said, it was probably an amplification site, but there were clearly patients who had antibodies to this virus before the big outbreak at the Wuhan wet market. And in fact, it wouldn't surprise me if this virus actually originated in southern China before it made its way to Wuhan, which is more centrally located in China, where it then started the human-human transmission event. So with that, I think I will stop talking, and I will stop my sharing if I can, and hand it back over.
Thank you, Tony, and thank you, Brian. We've got a ton of questions here, so I'm going to try to just dive right into some of our viewer questions. We're not going to get to all of them, but let me start with a question we had from Donna, which I actually think is fascinating, and we started to touch on it a little bit with Brian with the adaptation and evolution piece, but she was wondering, why do most illnesses seem to start in animals before humans? You know, so almost kind of like the reverse, is the human spreading the viruses to animals. Why does it seem like mostly we're seeing these come out from animals? Brian, do you want to start with that?
Well, you mean why do they mostly come from animals and get into humans? Because humans are a single species, and all the other animals other than us, there's thousands and thousands and thousands of those species. And so they all have their own stuff. What we have many of our own viruses and many of our own pathogens. We know about them. We don't know about all the crazy pangolins out there and the various species of pangolins or various species of bats and all their particular viruses. And so it's just a matter of numbers, really. Does that kind of make sense?
Yeah, it does. Tony, maybe build on that. And I think, and you can build on this thought too,
so
about the challenging of studying animals versus humans.
So humans we've done a really good job of controlling most infectious diseases, either through use of antibiotics or vaccinations. You know, if you go back to the turn of the 1900s, the early 1900s, only about 1 in 5 or 6 or 7 children in New York City survived to adulthood, because many of those kids, 10 or 15% of them would die from infectious diseases such as pertussis, whooping cough and measles. And now we've done a great job with antibiotics and vaccines to control those. And so it's not that we don't have our viruses and bacteria, we've just done a really good job of controlling them for the most part. We occasionally see outbreaks that tend to occur because of communities that don't, for example, vaccinate against measles. And that's the main reason why we've been able to control these. There's also sanitation and hygiene practices that have also minimized infectious diseases. So our control of infectious diseases, and if you look at the history of humanity, this is a very new thing, right? We've only been doing it pretty well for about 50 to 100 years. Now, as Brian said, there are literally, millions of species of animals out there, and we're just one of them. And some of the biggest drivers are, number one, do the animals encroach in human habitat, or do humans encroach on animal habitat, which is becoming more and more common, right? 100 years ago, you didn't see all these housing developments up in the mountains of Colorado like you do now. Now they're all over the place. If you look at the Nipah virus outbreak in Malaysia, the first one that occurred, it was because that abattoir where those swine were being kept was expanding into bat habitat. And so that brought the swine into contact with the bats. And so all of these events conspire to make these statistically unlikely events become more likely. And that's what's leading to these spillovers. And there's no change that's occurring. People aren't there. We're still not having the policies necessary to sort of put these barriers between wildlife and humans in many parts of the world. And so consequently, this is going to be an ongoing and persistent threat. So if there are any young people out there who are interested in infectious diseases, I think that's a great career to go in, because there's going to be a lot of job security for you in the coming decades. So.
Thank you. We're going to be crouching up on our time here, so I want to be respectful of your time and our participants' times. But why don't we end with, we had a couple questions from a couple different viewers that were along the same lines of what do you think is that dramatic or drastic step that we could use to help alleviate some of this spread, right? We are becoming a very mobile society. You're talking about the encroachment. So are there any, from both of your perspectives, and I'll start with Tony and then we'll end with Brian, any dramatic step that you think we should be doing or currently doing or being aware of, or potentially, if you want to get into the policy world too, a policy action?
So I'll talk about COVID-19, and maybe Brian can talk more generally because he does this as part of his job. But I think it's clear that if you, number one, wear masks, if you just get 95% of people to wear them, right? And you keep that 6 feet social distance, you limit the size of groups that you get in, still maintaining the mask and the social distancing. And distancing, right, isn't just about space, it's also about time. So I have some of my students come in during the day and others come in during the evening. And so we separate them through time, not physical distance. Try to make sure you wash your hands on a regular basis, and avoid these practices that sort of facilitate viral transmission. Then you can get this under control. We wear masks not to protect ourselves. We wear masks to protect others, right? Because what we're trying to do is, if you're infected and you're asymptomatic, if you have a mask on, you reduce the droplet distance that the virus needs to go by 6 feet or less. It's actually more like 3.5 to 4 feet or 5 feet that those droplets can go, and then they fall to the ground, and that really knocks it down. You put a mask on, and now that droplet distance is even much less. So what we're trying to do is knock down the ability of the virus to transmit from person to person, what we call the R0 number. And if you use all these things, you can reduce it to below 1. And if you get that R0 below 1, then eventually the virus will go away.
From a broad standpoint, I think it's multifactorial. We need to broadly try to do what Tony was saying and keep wild areas wild. That doesn't mean we can't visit them, because we need to visit them to develop the capacity to understand how beautiful and important sustainable wild lands are for human societies. But we need to allow them to grow and develop and not encroach so much. We also probably need to put in some policy changes on some very specific habits, like wet markets are probably not the greatest thing to have, where you're slaughtering a wild animal that's on top of cages of other wild animals. China had already talked about getting rid of those. They are also common in Indonesia and many other areas. Those are probably should be avoided. And then lastly, we really need to develop and fund modeling, and probably even more important, surveillance efforts amongst wild and domesticated animals to watch where these spillovers and outbreaks occur, and to get a handle on when they're occurring so that we can prepare to intervene and stop them before the spillover really emerges and the outbreak turns into an epidemic, which turns into a pandemic. At the tail end of that, a late phase response, certainly developing our public health measures so that we're ready to go with very clear measures, contact tracing and all the stuff Tony just talked about, and quarantine, but also some good countermeasures that the biopharmaceutical industry can make. Broad-based vaccines that can target broad classes of coronaviruses or henipaviruses or other things that are ready to go in our stockpile that we can quickly pull out of the freezer, grow up a lot of them. And even if they're not perfectly adapted to that one weird coronavirus that you didn't know about, they might be good enough to really cut down the disease if you could, and the spread of the disease, more importantly, if you can get them implemented very quickly. And therapeutics, drugs and other things like that.
Great, thank you both. Extremely informative. We did not do justice to all the million questions that we had for you. But thank you everyone for tuning in today. A huge thank you to Dr. Brian Foy and Dr. Tony Schounts from CSC for joining us. We do a written recap of all those, of all these recordings in addition to the videos as a reminder to our guests. If our guests have a few times, we may propose some of those additional questions in the write-up that they can answer for you if they're willing. So don't hold us to that, but we'll see how that goes. As a reminder to everyone tuning in, you can find all of that written material and video recordings on the Institute website. We have a nice landing page. Just go to institute.dmns.org. You can find everything from the past 12 episodes. This one was number 13. We hope you'll join us back here next Monday morning where we're going to be talking about mental health under this COVID-19 pandemic in our communities. So thank you for tuning in. A huge thank you to our partners at the Colorado School of Public Health and the Denver Museum of Nature and Science. As a reminder, follow us on Twitter at Institute SciPaul. You can follow our museum colleagues at Denver Museum NS and our School of Public Health at Colorado SPH. We appreciate your time dedicated to us this morning. So thank you and hope you have a great rest of your Monday and week. We'll see you next week.